Now we know that where is the chromosome present? Tell me. Where is the chromosome present? Say. I mean, inside the cell, where is the chromosome present? In the nucleus, right?
Yes, ma'am.
Hmm. See, inside the cell, there is the cytoplasm, and in that, there is the nucleus. Now, inside the nucleus, chromosomes are present, okay? And the DNA that we talk about, the DNA, our inheritance, or the genetic material, it is actually present on top of the chromosomes. So, chromosomes, you can call them as the physical carriers of the DNA, sort of like they carry the DNA. Now, Mendel started his work on the pea plant in 1865. Imagine, 1865. But no one knew about his work, no one understood his work until 1900. Okay, now why? What was the reason? Because he is now known as the father of genetics. Then why was his work not recognized? Okay, let us see. Mendel's work went unnoticed because of the following reasons. Now, up until now, you have learned about the law of inheritance, right? And
Yes.
you have learned about the cross, the dihybrid, the monohybrid cross. So, what
Yes.
is so unique that you found in it? Like, what is the one thing that you found different in all of the things that you have studied? Anything you found particularly difficult? Or, sorry, different? Say, I mean, I'm just casually talking to you. I'm not asking you a bookish question. It's just a
Mm,
casual question. Tell me, what is that?
no.
Nothing. You found nothing different. See, didn't you notice that he used numbers in biology? Up until
Yes.
now, whatever chapters you have read or studied or I have taught you, have you ever seen me using biology or the numbers or the ratio in it?
No.
No. But he was the first person to actually use mathematics in the field of biology. So this was the first and the most, you can say, one of the most great reasons that his work was unrecognized. Because no one at that point of time believed that, okay, maths and biology, they can't be correlated. That is why also, okay. Apart from that, during the 1800s, the communication was not that good, right? Just like how we have journals, right? Journals for the publication, then there are magazines, scientific magazines, and newspaper and everything. At that point of time, the communication was very minimal. So the lack of communication is the reason, is like one of the major reason that his work could not get widely published. Apart from that, he used mathematical and statistical approach in the field of biology, and that was unacceptable. Like, people did not understood that, okay, biology may be used in the field of like you can say maths and statistics may be used in the field of biology. No one agreed to that. Apart from that, he could not provide experimental proof for the existence of those factors. He said that genes are responsible for the inheritance of character, but he did not give any experimental proof for it. So people were like, okay, if you don't have proof, how can we believe you? Apart from that, chromosomes were not discovered at his time because microscope was not discovered at his time. Okay, so that is why the meiosis, mitosis, the different kinds of cell division, that was also not discovered. And the chromosomes were also not discovered at the time of Mendel's work. Now, in the 1900s, three scientists, that is De Vries, Correns, and von Scherma, now they independently rediscovered Mendel's result on the inheritance of characters. Now, these three scientists, they were working separately, and each of them came on to one conclusion that Mendel had already given, right? So, by the 1902, the chromosomes were also discovered because the electron microscopy was become, you can say, very developed. Okay, so all of these things came after, you can say, so many years when Mendel worked. Now, Walter Sutton and Theodore Boveri, there were two scientists, they noted the behavior of chromosome. This was parallel to the behavior of genes. and they use the chromosome movement to explain Mendel's law. So they noticed one very, since the chromosome, the divisions were noticed. So Walton Sutter and Theodore Boveri, what they did, they noticed that okay, the way the chromosomes behave, same way the genes were also behaving. So they thought, okay, there may be some connection in between them. And according to Sutton and Boveri, the pairing and separation of a pair of chromosome would lead to the segregation of a pair of factors or genes. Meaning, if the genes are getting separated, if the genes are getting inherited, then that means the chromosome had also gotten separated and segregated. Clear? So
Yes,
knowledge of
ma'am.
the chromosomal segregation plus the Mendelian principles. So this is like a collaborative thing. And then that gives us the chromosomal theory of inheritance. This is what we know. Like chromosomal theory, inheritance, we already know that inheritance takes place because of the genes, right? Certain genes that are there. Now, these inheritance, how is the inheritance happening? It is happening because of the chromosome, because the chromosomes are the ones that are carrying the gene. So if the genes are getting inherited, then that means the chromosomes are also getting inherited. So all in all, this is the chromosomal theory of inheritance. Clear?
Yes, ma'am.
Any doubts?
No.
Now, let us look at the features of the chromosomal theory of inheritance. Like the hereditary traits, meaning like the genes, the chromosomes retain their number, their structure, and their individuality. Meaning, for example, there is a certain chromosome that is being passed on from one generation to another generation. It does not mean that the number, the shape, the structure of the chromosome will change in the offspring. It will stay fixed just like how it is. And not just one generation, several generations also, the structure, the size, the individuality of the chromosome will stay fixed. Clear? So they will neither get lost nor do they get mixed up. So this is the first thing about chromosome. Then both the chromosome as well as genes, they occur in pairs in the somatic cells, meaning the body cells of the organism. So inside the body cells, the chromosomes and genes, they are present in paired form, paired structure, right? How many chromosomes do us humans have in total? Hmm?
26.
How many?
26.
26? No.
No, no, sorry.
Think. You are close. The number is in 20s, but not 26. 23 pairs. Okay. We have 23 pairs. Means how many in total? We have 46 chromosomes in our body. Clear? Now see, in
Yes,
our
ma'am.
body, I'll tell you one thing. For example, the cells that are present in our body, they are of two types. There are somatic cells. Okay, there are somatic cells. And then there are germ cells. Somatic cells, these are the body cells. Okay. And the germ cells, these are the sex cells, meaning which will give rise to the gametes. Clear? And these are the normal body cells. So out of the 23 pairs, 22 of them are body cells and one pair is of germ cells. Clear?
Yes, ma'am.
Any doubts?
No.
Why did you switch off your video? You got ashamed.
No, ma'am.
Then switch on your video. This much is clear?
Yes, ma'am.
See, if you answer something wrong, it's all right. It's not the end of the world. Okay, so don't feel that way. I am here to teach you. If you are wrong, I'll teach you. If you are wrong 100 times, I'll teach you 100 times. So don't be scared. Okay, and speak in the class. Whatever comes to your mind. I don't think that it is possible that someone would have no doubts at all. Isn't it? You have doubts, right? When you study, when you think of a topic, You have doubts, isn't it?
Yes.
So talk to me in the class. Don't be like that. Okay. So this much is clear.
Yes, ma'am.
Hmm. Now, the two alleles of a gene pair, they are located on the homologous sites on the homologous chromosomes. Right? See, homologous chromosomes are the ones that have the same structure, right? And they are located on the homologous sites. Now, both chromosome and genes, they segregate at the time of gamete formation such that only one of the each pair is transmitted to the gamete. We know that what is the ploidy of gametes? Gametes are haploid or diploid?
Haploid.
Very good. The gametes are haploid. Now, always remember that the gametes are haploid and the body cells, they are diploid, meaning 2N. So now when the gamete formation takes place, if you might remember human reproduction chapter, in that you've studied that chapter with me, right? Human reproduction.
Yes, ma'am.
So in that chapter, if you might remember like the gametogenesis part that we were studying, in that we saw that the gametes will always be haploid. So this is what this line is saying, that only, like for example, 22 plus X or Y. So either the X will go into the gamete or the Y will go. Only one of it will go into the gamete. And we know that gamete is formed by meiotic division. So this was all in all the chromosomal theory of inheritance. This is theory. Okay. Is this whole theory clear to you?
Yes, ma'am.
Hmm. Now, as I told you that this is just theory. So the proof for this theory was still yet to be found, and which was given by T.H. Morgan, Thomas Hunt Morgan. Now he gave the experimental verification. He verified the chromosomal of theory of inheritance experimentally. Okay, and how did he do so? He worked on Drosophila melanogaster. It is very important to remember. Thomas Hunt Morgan, he worked on Drosophila melanogaster, which is very commonly known as fruit fly. Now again, just like how Mendel chose which plant for his experiment?
Pea plant.
What is the scientific name of pea plant? Hmm? The initials are P and S. See?
I don't remember.
Pisum sativum. You remember
Yes,
that,
ma'am.
right? Pisum sativum.
Yes.
Hmm. So just like how Mendel worked with that, same way, Thomas Morgan, he worked with Drosophila melanogaster, but now this is not a plant, it is an insect. It is an insect which is a fruit fly. Now, why did he choose Drosophila? So the reason for him choosing the Drosophila was, firstly, the Drosophila can be grown in the lab. It does not require the, you can say, the real surroundings and everything. You can synthetically grow the Drosophila in the lab. Now, the second reason is that the fly, this particular fly, it has a short lifespan, meaning only it lives for 2 weeks and then it dies. That is it. Then a single mating produces hundreds of offsprings. Hundreds of offsprings mean hundreds of observations. So that is why it is very important. Now, you can easily distinguish the male and the female. Sexual dimorphism is there, meaning the male looks different, the female looks different. So it is easier for the experimentalist to see. Now, it has a very small number of chromosomes. The Drosophila has only 4 pairs of chromosomes. Just like how us humans, we have how many pairs of chromosomes? Say,
23.
yeah, 23 pairs of chromosomes. But Drosophila just has 4. So because of all of these reasons, it was his best choice to choose Drosophila for his experiment. Clear?
Yes, ma'am.
All right. Now you can note it down till here. Then we'll move forward. You can switch off your video, write down comfortably.
Done, ma'am. 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 Done, ma'am.
All right. Any doubts up until now?
No, ma'am.
Okay. Okay, now moving forward, we'll talk about the linkage and recombination. Now see, linkage and recombination, they are studied together because they were observed together. But they are very different things from each other. The way, or you can say how tightly the genes are located on any chromosome, that is what is known as linkage. Link means how tight is it? How tight is the bonding of the gene on the chromosome? Whereas when we talk about recombination, if you remember from class 11, if you've studied cell cycle and division, in that you might have seen that in the case of meiosis, there is crossing over that takes place. So that crossing over is what is called as recombination. So the generation of non-parental gene combination is called as recombination. And when does it take place? So it takes place during the crossing over process. Now, the sex-linked genes, they were studied by Morgan and obviously in the case of Drosophila. So he studied 4 pairs of chromosome in totality, out of which 3 pairs were autosomes. See, autosome is another word for somatic cells or body cells. And allosome is the name for sex chromosome. Clear?
Yes, ma'am.
So he carried out the experiment in Drosophila and he crossed out the yellow bodied, white eyed female with the brown bodied and red eyed male. Okay, the yellow color body with white eye female, brown color body with red eye male. And he saw that the ratio, it was not 9 is to 3 is to 3 is to 1, which it was supposed to be because it was a dihybrid cross, but it did not follow the Mendelian ratio. So what happens that if just in case the genes are so tightly linked on the chromosome, so there might be a case wherein the genes will not get segregated, or you can say they will not get crossover. So if the genes are tightly linked, they show very low recombination. And if the genes are loosely linked, if they are loose, so they can displace from one place to another. Right. So in the case when the genes are tightly linked, so the percentage of recombination is just 1.3%. But when the genes are loosely linked, then the recombination is seen in 37.2%. Now, Alfred Sturtevant, he used the recombination frequencies to measure the distances between genes and map their functions on the chromosome. So, Alfred Sturtevant, he did gene mapping. He wanted to see what the distance in between two genes were, how the genes were located, what their position was like, and he did the mapping of the chromosome. Now, just like how we use certain symbols or SI unit for whenever any kind of calculation or anything is done, same way for mapping also certain units are used. In the case of prokaryotes, the unit that is used is map unit. And in the case of eukaryote, the unit that is used is centimorgan. Okay. Now, the crossing over, you already know that it is the exchange of genetic material. It is happening during the crossing over process. And for example, if there is 1% crossing over that occurred, So if in terms of prokaryotes, we will call it as one map unit. Clear?
Yes, ma'am.
Now then there is the sex determination. Now sex determination is basically what kind of genes would get inside the gametes of any individual which will make them a male or a female. But it's not just always the genes that will determine the sex of the offspring. Establishment of the sex of an individual through different ways, right? Through any ways. It can be related to the chromosomal pattern, related to the environmental pattern. So it can be any way. And this is what is called as sex determination. Now, in the case of turtles, the sex is determined by the incubation temperature. For example, how long did they get the temperature, how less did they get, that determines their sex. But in the case of Drosophila, in the case of birds, humans, butterflies, grasshoppers, and honeybees, the sex is determined by the genes. The genes are responsible for the determination of sex. Clear? All right. Now you can note it down till here. Then we'll move forward.
Done, ma'am. Done, ma'am.